Premature birth disrupts normal lung development. Prolonged exposure to hyperoxia during this critical period results in decreased cell proliferation and altered metabolism, which can lead to developmental arrest, simplified alveolarization, and dysmorphic vessels, as seen in bronchopulmonary dysplasia. Recently, genes involved in the generation and regulation of the circadian rhythm have been found to modulate cell proliferation and metabolism. It is not known whether these are involved in the pathophysiology of BPD in the developing lung. In the mouse, chronic jet lag results in circadian disruption and alterations in clock gene expression and lung mechanics. Circadian rhythmicity is driven by the master clock in the suprachiasmatic nuclei of the hypothalamus. The SCN integrates information from light and other environmental cues and relays them to various peripheral clocks in the body, thereby controlling their function. A peripheral clock exists in the lung. At the molecular level, in the lung and other tissues, circadian generation exists as multi-level hierarchical regulatory loops. The clock B-mel heterodimer activates transcription of HER and CRY genes via E-boxes in a promoter. HER and CRY proteins translocate to the nucleus and form a repressive complex, which inhibits clock B-mel transcriptional activity. Hence, HER and CRY shut down their own transcription. This allows a new cycle to start. Additionally, clock and B-mel activates reverb transcription, resulting in daily fluctuations in reverb. When reverb is bouncing its natural ligand, reverb represses the circadian rhythm, inflammation, metabolism, and its own expression. In the NICU, premature neonates are frequently exposed to disrupted light-dark cycles as well as to oxygen therapy. However, not much is known about the combined effects of these two stressors on the developing lung or on circadian gene expression. Therefore, we hypothesize that in neonatal mice exposed to hyperoxia and allowed to recover in room air, circadian dysregulation results in worsened lung injury and altered circadian gene expression. The objectives of this study were to determine the effects of acute hyperoxic exposure and recovery on the expression of key circadian genes, and also to determine if disrupted circadian rhythm worsens neonatal lung injury caused by hyperoxia and recovery. Towards this objective, neonatal mouse pups less than 12 hours old were exposed to either constant light or 12-hour light-dark cycling, as well as to either room air or hyperoxia. Circadian rhythm was disrupted by exposure to continuous light, designated LL. Hyperoxia was achieved by exposure to 95% oxygen in a specifically designed chamber, designated O2. Controls were exposed to 12-hour light-dark cycling in room air, designated 12H-LD/RA. Following a 3-day exposure, the groups were recovered in room air under 12-hour light-dark cycling until postnatal day 14. On day 14, the pups were sacrificed and their lungs were dissected for further analysis. Steady-state mRNA levels of key circadian genes and markers of vessel development were evaluated using quantitative PCR. Additionally, immunohistochemistry for von Willebrand factor and alpha smooth muscle actin were performed to evaluate pulmonary vessel density and pulmonary vessel wall thickness, respectively. Lung architecture was examined by H&E staining of lung slides, by radioalveolar counts, and by alveolar wall thickness measurements. These graphs represent steady-state mRNA levels of key circadian genes in the lung. The dark blue bar represents room air and 12-hour light-dark cycling. The light blue bar represents room air and constant light. The red bar represents hyperoxia and 12-hour light-dark cycling. And the pink bar represents hyperoxia and constant light. There were no statistically significant differences in reverb alpha or B-mel expression on day 14. However, HER1 expression was decreased in the presence of hyperoxia and constant light. Next, pulmonary vascular markers were evaluated. Both FLT1 and TIE1 are precursors to vascular development and are under circadian control in the adult lung. At day 14, both FLT1 and TIE1 gene expression was significantly decreased in the presence of hyperoxia and constant light. Pulmonary vessel wall thickness was then evaluated. Lungs were stained with von Willebrand factor and co-stained with alpha smooth muscle actin. Von Willebrand factor is present in the endothelial cells that line the vessel walls, and alpha smooth muscle actin is present in the muscular walls. In the figure to the right, red represents alpha smooth muscle actin, green represents von Willebrand factor, and blue represents DAPI. To calculate the mean thickness index of a particular vessel, the area inside the external border of the alpha smooth muscle stain was measured, and this was subtracted by the area inside the internal border. This value was then divided by the area of the external border and multiplied by 100. Hyperoxia and constant light increased vessel wall thickness compared to all other groups. Thus, the combined insult of both stressors resulted in the thickest pulmonary vessel walls. Next, the density of the pulmonary vasculature was assessed. To evaluate vessel density, sections were stained for the presence of von Willebrand factor, and the number of von Willebrand factor positive vessels per high-power field under 20x objective were counted. Lungs exposed to hyperoxia and constant light showed a trend towards decreased vessel density, although these results were not significant, with only 2 liters tested thus far. Next, pulmonary architecture was studied. Radial alveolar count, an objective measure of alveolar number, was assessed. Hyperoxia and constant light had reduced radial alveolar counts, suggesting alveolar simplification. Next, lung structure was further assessed by examining alveolar wall thickness. To study alveolar wall thickness, H and E lung sections were viewed under a set number of horizontal lines, and the thickness of each septum was measured parallel to each intersecting line. Similar to the RAC data, hyperoxia and constant light resulted in thickened alveolar walls. These are the representative H and E lung sections at 14 days. Lungs from the hyperoxia and constant light group exhibit the most distorted alveolar structure. This is demonstrated by wide simple air spaces and thickened alveolar septal walls compared to the room air 12-hour light dark control group. The lungs of neonatal mice exposed to all other conditions closely resemble the control, suggesting that it is the combined insult of hyperoxia and constant light that perturbs alveolar development and alveolar wall thickness. To conclude, neonatal mice exposed to hyperoxia and constant light had decreased expression of circadian genes, decreased expression of vascular markers, thickened vessel walls, decreased vessel density, alveolar simplification, and thickened alveolar walls. Thus, by disrupting circadian rhythm during hyperactive exposure in neonates, this can lead to disruption of lung architecture and function. We speculate that neonates exposed to both oxygen and disrupted light-dark cycling, which often occurs in the NICU, may be at greater risk for developing BPD. I want to thank everyone in the lab for their help and support. This work is funded from a grant from the NHLBI.